Good morning, my dear students. Welcome to our Zoology class. Hope you are very happy. And in the last class, we studied about some one important topics about the fifth lesson molecular genetics, transcription, the process of mechanism of transcription. Okay, this is the very important 5 mark question. In the last class, we have studied. Okay, and some students send that answer to me. I really appreciate you. Then today we are going to study about three important topics. Okay, listen students. So listen very carefully. All these three topics are very important for 5 mark question. Okay. So the first topic is genetic code. Okay, the first topic is genetic code. One important 5 mark question is there. So listen very carefully. See, genetic codes means, see, in the previous classes already we have studied DNA is the genetic material. Okay, DNA is the genetic material of maximum living organisms. Okay, and this DNA molecule only carrying the genetic informations in a cell and carrying or transferring the genetic information from one generation to next generation. Okay, so carrying the genetic information in a cell and carrying the genetic information from one generation to, or transferring the genetic information from one generation to next generation. Understand a few? Then inside in this DNA molecule, these genetic informations are written in a language or written in a particular code. Okay, and maximum these codes are in triplet forms. Okay, so these codons. Okay, these codes are represented by some letters. Okay, capital letters, and three letters are togetherly forming one codon. So three letters are present in one codon, and each and every letters are called code. Okay, so inside the DNA molecule, the genetic informations are stored in a code, and these three codes are togetherly forming a single codon. So the codons are maximum in triplet forms. Okay, triplet forms meaning three codes are togetherly forming a single codon. Hope you understood the meaning of this genetic code. Understand? Okay, now we are going to study about the first important 5 mark question, the salient features of genetic code. Listen very carefully. See, here I have written 9 important salient features for this genetic code. The first one is 64 codons. Totally 64 codons present in the cell. Okay, totally 64 codons are present. In these 64 codons, 61 codons are code for amino acids. Okay, so 61 codons are playing a very important role in the protein synthesis. Understand? Then the remaining 3 codons are responsible for stop the function. Okay, stop the protein synthesis. Understand a few? So totally 64 codons are there. The 61 codons are responsible or code for the amino acid, and last 3 codons are called stop codons. Is stop the process. Then second one is universal. This genetic code is universal. That means in all the living organisms, maximum living organisms, the genetic code is same. Understand a few? Understand a few? So, universal meaning the genetic codes are same for all the living organisms. For example, see here, U. This is one codon. This codon code for one amino acid called phenylalanine. In all the organisms, every organism, this codon is code for this phenylalanine. But some exceptions are there. In prokaryotes, these codons, okay, these codons are involved in the chloroplast production. Understand a few? So the second point, the second salient features of the genetic code is universal. The triplet genetic code is same for all the living organisms. Then third one is non-overlapping codon. Here non-overlapping codon meaning the same letters should not be used for two different proteins. Okay, two different codons. Understand a few? So same letters. Okay, same letters should not used for two different codons. So this is the meaning of this non-overlapping codon. The third one is commaless codon. That means, say, this is one of the very important characters. Say, here I have written three letters. Already I told you this is one chord, second chord, third chord. These three chords are togetherly forming a single chord. So, in between two chords, we should not put any punctuation. Full stop or comma, anything. We should not put anything in between these two chords. It should be directly read from one end to next end. So, this character is called commaless. Then, next one is degenerate code. Degenerate code means, say, more codons code for a single amino acid. For example, see GUC, GUA, GUG. Okay, all these three are different codons, and these three codons are code for the single amino acid valine. Then, next one is non-ambiguous code. That means single codon code for single amino acid. Okay, that's the meaning of this non-ambiguous codons. And the next, seventh point is, see, this codon should be read from one end to another end. That means it should be read from 5 end to 3 end. Already I told you two ends are there. One end is called 5 end and the second one is called 3 end. Okay, so in the codon should be read from the 5 end to 3 end. Then next point, the 8th point is dual function. Some codons doing two functions in a cell. For example, see AUG. This AUG codon start this process. So it acts as a initiator codon, and this codon also code for one amino acid called methionine. So dual function meaning two functions. Okay, some codons doing two important functions. For example, this AUG is act as a initiator codon and also code for methionine. And the last silent feature is, already I told you, 64 codons, 61 codons code for amino acid, and 3 codons are stop codons. See, these three are the stop codons. UAA, UAG and UGA. These three codons are stop codons. Understand a few? So now we have completed one important 5-mark question, salient features of genetic code. Hope you understood that. The next 5-mark question is mutation. See, mutation means generally we can say sudden change in the gene. Okay, sudden change in the genes is called mutation. See, changes in the nucleotide. Okay, changes in a nucleotide that substitute one for another one base. Okay, one base for another one base. Okay, this condition is called mutation. This condition is called mutation. Okay, so this mutation leads to many problems, many disorders, some changes in their characters. Okay, so these are the problems because of the mutation or because of the changes in the genes. Okay, so now we are going to study about one important disorder due to gene mutation. Name of that disorder is called sickle cell anemia. Okay, sickle cell anemia is one of the very dangerous disease affecting the RBCs of the human beings. Okay, normally the shape of the RBC is circular shape. Okay, round shape and biconcave in nature. If anyone is affected with this disorder, sickle cell anemia disorder, the RBC is in sickle shape. RBC is converted into sickle cell. So it is very important. It is a very dangerous disease, and if anyone is affected with this disorder, the oxygen carrying capacity of the blood is totally reduced. Understand a few? Then listen very carefully. Here, the hemoglobin molecule. It is one of the very important pigment present in the RBC, giving red color to the blood, and this hemoglobin pigment only carrying the oxygen. Okay, carrying the oxygen from one place to another place. Understand? And this hemoglobin molecule is made up of 4 polypeptide chains, 2 alpha chain and 2 beta chain. And both chain, alpha chain and both chain carrying a heme molecule also. This heme molecule only binding with the oxygen. Understand a few? And the genes responsible for the production of this hemoglobin, normal hemoglobin, is called beta hemoglobin gene. Okay, so the alleles present in the beta hemoglobin gene is involved in sudden change or involved in mutation. Okay, so if this gene is involved in mutation, it cannot produce normal hemoglobin molecule. The affected genes or the genes involved in the mutation can produce only abnormal hemoglobin molecules. Understand a few? Understand a few? Then, see, in the sixth position of this beta hemoglobin gene, one codon is there. Name of that codon is called GAG. GAG. Understand? So this codon is present in the sixth position of a beta hemoglobin gene. Understand a few? When this gene is involved in mutation, this codon is changed into another one codon. Name of that codon is called GTC codon. GTC codon. Hope you understood. So beta alleles present in the beta hemoglobin gene only responsible for the production of normal hemoglobin molecule. When this gene is involved in mutation, in the sixth position, the codon GAG is changed into GTC codon. So it cannot produce the normal hemoglobin molecule. Then this codon is responsible for the production of one amino acid. Okay, one amino acid. Name of that acid is called glutamic acid. So this codon is responsible for the production of glutamic acid. Already I told you, if when this gene is involved in mutation, this codon is changed into this GTC codon. So this GTC codon produce another one amino acid, valine. So instead of producing this glutamic acid, here what amino acid is produced? Valine. Understand? So instead of glutamic acid, valine is produced. So the hemoglobin is in abnormal condition. The hemoglobin is in abnormal condition. Hope you understood. So this is the reason for the sickle cell anemia. So in the normal beta globin gene, sixth position, the codon name is GAG codon is present. This codon is changed into GTC. So instead of this glutamic acid, valine is produced. So it leads to, as a result, some abnormal hemoglobin molecules are produced. So it leads to sickle cell anemia disorder. Hope you understood. Then see, when we insert or when we delete a small single code or double codes from a codon, the total arrangement is altered or changed. Listen very carefully. See, A, B, C, A, B, C one codon, then D, E, F second codon, G, H, I is the last codon. So it's a normal arrangement. When we insert one O in between these two codon, the total arrangement of the codon is changed. See, first already I told you it's a triplet form. The codon always will be in triplet form. So A, B, C, then D, E, F, then this O will come. Already we have inserted this O in between these two codons. So O, G, H and I is separated. Okay. Then if we add one Q also, what will happen? A, B, C, then D, E, F, then what? O, Q, G, then H, I. Like that the codon's nature is changed. Understand a few? So when one code is inserted or deleted from a codon, it leads to some changes in the entire codons. Understand a few? Understand a few? Then one important 2 mark question. See, the addition or deletion in a base change the reading frame from one end. This condition is called frame shift, insertion or deletion mutation. So when we add one code or when we delete one code, totally the reading, the frame is what changed. The frame is changed from one unit to another unit. So this condition is called frame shift insertion or deletion mutation. Or is called a deletion mutation. So it's one of the important 2-mark questions. Understand a few? So now we completed 2 important 5-mark questions. First question, salient features of genetic code, and the second question, mutation. Hope you understood. Then the third question is, now we are going to study about the third question, structure of tRNA. tRNA is also called adapter molecule. Okay, tRNA is also called adapter molecule. See, in this chart I have drawn the structure of tRNA. Okay, see, the tRNA is also called adapter molecule because of 2 reasons. The first reason is this tRNA molecules only picks up or carrying the amino acid scattered inside the cytoplasm of the cell. Okay, so the tRNA molecule, T means here transfer. Okay, so the tRNA molecule picks up or carrying the amino acids which are scattered in the cytoplasm of a cell, and this tRNA also coded for mRNA, so that only this tRNA is also known as adapter molecule. Understand a few? Then second point about this tRNA is, see, the 2-dimensional structure of this tRNA molecule was first proposed by Robert Coley. So Robert Coley is a scientist. He only first discovered or proposed the 2-dimensional structure of this tRNA molecule. Understand a few? Then this tRNA molecule is look like a clover leaf. And it is also look like a inverted L shape. Understand? So now I said 3 important points. Then this tRNA molecule has 4 arms. So this, the first arm called DHU arm, second arm is TC arm, then third arm is called middle arm, and in between this middle arm and TC arm, one more arm is there, is called extra arms. Okay, so the tRNA molecule has 4 arms: DHU arms, TC arms, middle arms, and extra arms. Understand? Then at the end of the arms, loop-like structures are there. Okay, there are 3 loops are present in these arms. The first loop is called amino acid binding loop. First loop is called amino acid binding loop, and the second loop is called anticodon loop, and the third one is ribosomal binding loop. So amino acid binding loop, anticodon loop, and ribosomal binding. So these are the 3 loops present in the arms of the tRNA. Hope you understood. Then next point, see, this tRNA, see here, the movement between the codon, already I told you, see, this is the maximum the amino acid molecules are attached in this region. Okay, so the movement in this region, then this region is called anticodon region. 3 anticodons are present in this region. Okay, so the movement of amino acid, the movement between the codons and anticodon makes a tRNA code for many amino acids. Okay, code for many codons. Okay, code for many codons. Understand a few? Then already I told you this region is called anticodon is there. Okay, anticodon is 3 anticodons are there. These anticodons are code on mRNA also. These 3 anticodons are code for mRNA also. Understand a few? Then next point. See, Here in this end, the amino acid molecules are added one by one. The amino acid molecules are added by a process. It is called amino acylation. It's called amino. So the process of adding the amino acid in this region is called amino acylation. Now this tRNA is called amino acyl tRNA or charged tRNA. After adding many amino acids, the tRNA molecule is called amino acyl tRNA or charged tRNA. And the tRNA molecules without this amino acylation is called uncharged amino tRNA. Is called uncharged tRNA. Hope you understood? That say, already I told you amino acylation. Amino acylation means the process of adding the amino acid in the tRNA molecule. Okay, and this amino acylation process is controlled by one enzyme. Name of that enzyme is called amino acyl tRNA synthetase. Amino acyl tRNA synthetase. So this amino acyl tRNA synthetase enzyme only responsible for adding amino acid in the tRNA molecule. There are 20 amino acyl synthetase are still discovered. Okay, 20 amino acyl tRNA synthetase is identified. Hope you understood. Then next point about this tRNA molecule is same. When this is one tRNA, when two tRNAs brought together or comes closer, immediately peptide bonds are formed. Immediately peptide bonds are formed. And the amino acid molecules inside a polypeptide chain also joined or also connected with peptide bond. So when two tRNA molecules coming closer, immediately peptide bond is formed, and the amino acid molecules present in a polypeptide chain also joined with the peptide bond. So peptide bond playing a very important role in the structure of the tRNA molecule. Understand if you understand? Then say some tRNA molecule, okay, code the informations on the mRNA molecule. Okay. Some tRNA molecules code the information on the mRNA molecules. And this tRNA molecule has some complementary bases. Okay, complementary bases, and these complementary bases are suitable for mRNA. These complementary bases are suitable for mRNA. Hope you understood? Then the last point about this tRNA is, say, some tRNA molecules are specific for initiation process. Okay, so for initiation purpose or initiation process, tRNA molecules are present, but no tRNA molecules for stop codon. Okay, so for initiation process, tRNA molecules are there, but to stop the process, no tRNA molecules. Understand a few? So now we have completed the third 5-mark question, very important, and this diagram is very important for 2-mark question. Understand a few? Understand? So dear students, today we have completed 3 important 5-mark questions and 1 or 2-mark questions also, and many 1-mark questions also there. So today itself try to complete these 3 questions. All these 3 questions are very small questions, you can easily understand. Study well and send the answer to me through WhatsApp. Okay, my dear students, I hope you will set. Okay, thank you. We'll meet in the next class.